Total Oxidant Status Colorimetric Assay Kit Operation Guide. Before the experiment, check all kit contents before use. Contact us if needed. Sample preparation. If the sample is turbid, it is recommended to centrifuge at low temperature and test the supernatant. Reagent preparation. Prepare standard dilutions with double distilled water according to the recommended concentration gradient. Then vortex to mix. Operating steps. Add 20 microliters of standard solution with different concentration to the standard well. Add 20 microliters of sample to the sample well. Add 200 microliters of chromogenic agent to each well. Measure the OD values of each well at 590 nanometers with microblet reader, recorded as A1. Add 50 microliters of substrate to each well. Incubate at 37 degrees Celsius for 5 minutes. Measure the OD values of each well at 590 nm with microblet reader, recorded as A2. Data processing. Export data and calculate results as instructed in the manual.